G1 is the stage in which the cell grows and is at the point when most cellular activity happens. Now the cell moves on to the S stage, in which all cellular matter duplicates. In the last stage of interphase called G2, the cell continues to grow, preparing for cell division. Finally, the cell is ready to start mitosis. The first stage in mitosis is called prophase. The chromatin condenses into chromosomes. During this time, the centrosomes are creating mitotic fibers. Now the cell is entering prometaphase, so the nuclear membrane starts to break down and the chromosomes start to form. The black spots are called kinetochores. Then, the mitotic spindle begins to attach to the newly formed kinetochores. The cell then enters metaphase. The mitotic fibers enter the centromeres on the metaphase plates, preparing for anaphase. In anaphase, the two centromeres of each chromosome come apart, separating the sister chromatids. Once they are separated, they are considered a daughter chromosome. The spindles continue to separate the chromosomes as they shorten. As the chromosomes are pulled farther apart, the cell starts to elongate, preparing for division. Now, the cell is entering telophase, in which the cell continues to elongate. The chromosomes start to relax as the nuclear membrane starts to reappear and the DNA unwinds. Then, a cleavage furrow starts to appear so the cell can split into cytokinesis. Cytokinesis is when the cytoplasm in the cell divides, thus completing the cell cycle. The outcome of the cell cycle is the creation of two daughter cells. The purpose of this cycle is to create new cells to take the place of dead or damaged cells. Mutations in two basic classes of genes, proto-oncogenes and tumor suppressor genes, are what lead to cancer. Cancer is a complex group of diseases with many possible causes, like genetics, tobacco, over-sun exposure, and other carcinogens. The cell cycle is affected by the oncogenes and the tumor suppressor genes beginning to mutate in the synthesis stage.